Pessoal, bom dia, né? Nós temos então agora a nossa palestra de encerramento do evento. Antes eu vou apresentar um egresso da universidade, que é o professor Odair, que ele foi aluno de graduação, licenciatura aqui de Biologia, da UEM, algum tempinho, né, Odair? E foi orientado do professor Souza e realizou o seu mestrado e doutorado na Unesp. Eu vou passar a palavra para ele, porque ele conhece bastante o professor Hugo, que é o palestrante de hoje, realizou o seu estágio, né, com ele. Então, eu vou pedir para o Odair, né, conversar um pouquinho com vocês e apresentar o professor. Tá? Esqueci de falar que ele é professor da Unesp também, mas ele ia falar, né?
Obrigado, Lindamir. Bom dia a todos. Eu gostaria de agradecer muito ao professor Souza, à professora Lindamir, pelo convite de estar aqui participando desse evento. É muito gratificante, muito gostoso voltar aqui, né, como egresso. Eu gosto muito dessa universidade, então sempre é um motivo de muita alegria estar aqui. O professor Hugo, ele trabalha com sistemática vegetal e evolução. A família modelo dele tem sido a família Cactaceae nos últimos anos que ele tem trabalhado. A nossa colaboração começou em 2010, com parte do meu doutorado sanduíche, né? Eu fiz praticamente metade do meu doutorado lá, na Universidade de Saskatchewan, junto com o professor Hugo, e foi uma colaboração de muito sucesso, que a gente vem trabalhando junto até hoje, né? Ele, na década de 90, trabalhou bastante com a filogenia molecular, quando esses estudos estavam despontando bastante, né? Há alguns anos para cá ele mudou um pouco sua linha de pesquisa e tem vindo trabalhando com biologia reprodutiva das plantas. A palestra que ele vai dar é sobre biologia reprodutiva, né, das angiospermas, com exemplo nos modelos que ele vem estudando da família Cactaceae. E ele também vai tomar alguns minutinhos da palestra para apresentar a universidade para vocês e os programas de intercâmbio. O Canadá e o Brasil têm uma boa relação, né, acadêmica, de comércio. Então, a própria Aline, né, que também defendeu o doutorado dela aqui no PGB há pouco tempo, esteve lá com o sanduíche. Então, o Canadá é um país muito receptivo a todo mundo, boas conexões com o Brasil. Então, mais uma vez, obrigado.
Obrigado, grazie. Grazie, Dair. Bom dia, everybody. Me desculpa, pero meu português é muito ruim. Moro em Itália desde julho. Adesso a minha cabeça é um pouco complicada, as neuronas não funcionam bem. Não posso fazer a palestra em português. Eu vou falar em inglês. Primero que nada, gostaria de agradecer a los organizadores del evento, profesor Luis Souza y Lindamir, por la invitación, Odair, aunque sí por coordinar parte de la estructura del simposio, al Departamento de Posgraduación de la UEM, as well, por la invitación. Es siempre un placer para mí retornar aqui a Maringá, a Brasil. Eu comecei minhas colaborações em Brasil em 2004 com Lucia Loman, é uma amiga beníssima, queridíssima para mim. E ainda continuo colaborando com ele. Adesso, a minha apresentação é dividida em duas partes. A primeira parte the first part is in relation to the organization of the university. In general, I'm going to present an overview of the university very quick, and then I'm going to discuss or present the options that exist as students, as faculty, as PDS, as postdoctoral students, okay? The mobility of students and general personnel between the two universities. Perhaps you know some of those options, perhaps you do not, okay? And whenever these options come maybe once, twice a year, I'm sending out messages to people I know here in Brazil to see if there are somebody who is interested in continuing the education in the United States or getting at least some training in some areas of the area. But let me start. How many of you know where is Saskatoon? I'm coming from Saskatoon. Onde é Saskatoon? Conhece Saskatoon? Again? Saskatoon is centro de Canadá. It's in the center of Canada. It's right here. Es el cordón umbilical, prácticamente, de Canadá. Es una ciudad, a small city that is near the border with the United States, but it's a little bit isolated. The main cities in Canada are in this part, which is Montreal, Ontario, Quebec, and then BC and Calgary here. Everything else is lost somewhere there, okay? And so we are lost. So we are very far away from the rest of the activities, but it's a beautiful, beautiful place, okay? And the city is crossed by a river that we call the Saskatchewan River, South Saskatchewan. And the city, it has 11 bridges, and it's a little bit of the problem with the traffic as the city grows up. Crossing the river at the peak hours and the rush hour is complicated, but that doesn't take the beauty away from our landscape. It's very clean, very pristine, and the quality of life is outstanding. When I went there for job, I thought I was going for 2-3 years, and now I call it home. I love it too much. I thought I was going to go to Saskatoon, okay, then I moved to another place, more interesting. No, this is a very nice place. So, the city also offers one of the most spectacular Sundays or sunsets and daylights that exist or that you can actually experience in the prairies. The prairies, the praderas, are spectacular because you see a sky that is endless, endless, endless. And it's most of the time sunny, except when it's stormy. And the winter can be really, really, really rough because the temperatures can go beyond, even below -30, sometimes -40. We experience -40, right? And that is something that really, really can discourage a lot of people who are not used to cold weather. But nonetheless, it's not forever. When summer comes, everybody lives outside. We move outside. In the winter, we work inside, and we are more productive. And in the summer, we spend more time outdoors. Yeah, so that's the truth. So another beauty that we have, the city itself is gorgeous. It's small, around 250,000, and it's called the Paris of the Prairies because of the beauty that exists there. Okay? And we have the opportunity to see, particularly in the winter, these spectacular aurora borealis. Okay? Not every day, but especially when the atmosphere is in the best conditions, we have this opportunity to experience that spectacle. As per the university, our university is among the top 10 universities in our country. We have around 22,000 students, okay, of which 18,000 are undergraduate students and around 14,000 are graduate students, right? We have the only Canadian light source, you know, the synchrotron that is in Canada is located in Saskatoon. That has changed the life of Saskatoon in terms of science. So it's attracting a lot of scientific community, not only from Canada, but also from the world to do the research. So we are basically booming in many, many, many ways, okay, attracting the attention of the world. We have 14 colleges, okay, and perhaps I'm just going to mention some of those that are more related to my field, perhaps, but you can think about physics, chemistry, anything that you can think is there, okay, anthropology, et cetera, et cetera. So in the agriculture college, the most important programs or departments that exist are these: agricultural economics, animal science, crop science, and plant science, and soil science. In our college, I belong to the College of Art and Sciences, okay, and also a little bit to the agriculture, I should say, and I'll explain that later. The biology department is one of the largest, the one that has the largest amount of students. So we really do a very good job in attracting students, okay? and exist the other different departments. The university is 110 years old. Okay? And every single building that you see there, many of the new buildings look like they were built 100 years ago. So it's a very beautiful design that is being kept in the university. And it uses local material. The rocks are locally from the southwestern Saskatchewan to bring in for the construction of this sandstone that exists here. This is the observatorium that we have, and this is the new medical building that we have. This is the university, how it looks now. So it's fall there. Okay, it's all the opposite that you have here. So we're getting here to spring, right? And then we're getting back to this fall, and we're getting into to winter. So the trees are leafless. The grass is dying, okay? And eventually, things turn like this, turn white, okay? Sometimes the snow comes in as early as the end of October, okay? It goes, it can go. But if the temperature doesn't rise, depends on the amount of snow, can be 5–10 centimeters, it can melt in the next week or so. But then, if it doesn't raise the temperature, it stays. So we have a diversity of students, and the campus is spectacular. It's spectacular in many ways, not only because it has the different buildings that also keeps some of the old ancient buildings part of the experience for the education. And this is one of the ones, for instance, that we have, and the university is very strong in educating students, foreign students, in English, okay? These students of this program accept students that want to go to college education, okay, to have the minimum English requirement. So there are different levels from basics to advanced courses, and that will depend on your level, how much time you spend there before you get admitted to university. You are not admitted in an academic program in our institution without proficiency in English. I'm sorry to say that, okay? You can work on a personal basis with a professor and perhaps not have exactly 100% English, but still good communication skill, okay? So my lab is applied systematics, as I explained. I have been doing a whole lot of things since I moved out of Mexico in the late 80s and went to the States, and a master's, a PhD, a postdoc, and I went to Saskatchewan when I couldn't even pronounce Saskatchewan, you know. I saw the word Saskatchewan and I said, okay, is it in Russia or where is it? And so it told me, surprise, surprise, it was in Canada. So not far from when I was in Missouri Botanical Garden. My lab is very diverse and has always had, especially international students. They are the dominant, actually, the dominant. The Canadian are the minority and the international are the majority, which is a lot of fun because also those students, Canadian students, learn a whole lot from the different cultures that we have and become more open-minded and accepting diversity, particularly. I teach several classes at the undergrad level. One of the most, perhaps, popular classes is this one, the economic botany, okay, in which students learn about the origin of crops, the most modern crops or current crops that we use, how they came about, okay, and we do several exercises, hands-on, so that you understand how culture works, like from making, when we talk about corn domestication, we make tortillas. And if they want to make their own tortilla, they have to wear their hat. Otherwise, they cannot go and eat tortilla. Okay, so it's part of the deal. And we made sushi, we made beer. So it's super, super interesting course that the students like. It's perhaps one of the most popular courses in the university. I teach the general botany as well. It's a little bit more challenging, a large group. But unfortunately, like in most schools, botany is not the most popular topic, perhaps because all the terminology in part, you know, and in part also because of the professor. So I try to be more fun. I changed all the rules in my classrooms, and I teach in different ways, in such a way that the students connect and have more of the experience, and they remember things when they go home and after they finish the course. They don't memorize. I hate students memorizing. Okay? I like the students understanding the material, and then they can apply it when they leave the class. And they remember, that professor actually was right. So I like that. So we had that. This is a more advanced class, plant taxonomy, in other words. It's a very demanding class. It's a very small. I never get more than 15 students. The first class, the first week, there are like 25, 30 students there. And then when they learn that this is going to be very tough, they desert. Okay? And I end up with 12, 15, which is okay. But the ones that stay are really, really trained, which are actually the students that will follow, hopefully, some of the job that I'm doing. Okay? The training, that's the most important part for me, training students in the field that I love. Okay? And I have a whole lot of visiting scholars. Odair is here, with Choi, one of the Korea. Marcia was the first postdoc that I had from USP. Okay? She worked with, graduated with Professor Salatino, and she worked there and with Rich Salis and in seed morphology and molecular work. So Marcia stayed with me in 2 years and decided to stay in Canada because she did a good job and she continued to do that. And this is part of the field trips that we take, for instance, for the classes. Like here you go and you learn your techniques, okay? And it's part also of the connection that we have that I like having in my classes and with the students. I don't enjoy just going and be the boss, okay? I'm the chef, pero, and nobody talks. You know? I like to hear the conversation because I don't like monologues, okay? And so I like the interaction. The other part of my half, the other half of my job, I'm the curator of the herbarium in the plant sciences department, which I mentioned is in the College of Agriculture. So I'm a hybrid. In other words. Okay? And there we do all this type of research and botanical collections, digitization, biodiversity, vegetation, et cetera, of Saskatchewan flora and vegetation, right? We have most of the collection in our herbarium, which is about 200,000 specimens. 70% of those are from Saskatchewan. There is 30% remaining is from the rest of the world. So we specialize basically in prairie plants, okay, grasses. We have a lot and the best collection of grasses that exist in the prairies, all right? So Canada, in terms of biodiversity, is not very rich, okay? And you can imagine that because we are up in the north, okay? So we get very few days of growing conditions, okay? Propagation, et cetera, as opposed to here, that you basically can grow plants year-round, the whole year, okay? So our diversity is about 5,000 species, including all those introduced species, okay? For Canada. For Saskatchewan, only 1,400 species, which is nothing, okay? When we compare particularly with Brazil, all right? So I'm not gonna lecture in Brazil flora or anything like that because I know very little. I have worked with Lucy, as I mentioned before. I'm very fortunate to be in Anavillanas and areas of Manaus and the Amazon, but still know very little about the flora of Brazil, okay? And I've been lucky to work with Odair and Alini and learning something else about other tropical families, okay? So when we compare these two countries, you can see the diversity that you have as opposed to the diversity that we have, okay? It's minimal, almost nothing compared to what you have here. So that's what we do in the herbarium. My teaching has been good, has given me quite a few awards, so I'm very happy with that, so I can retire happy and not frustrated, and aposentar contento, eh? See? So anyway, as per the opportunities that exist in Canada, there are many, but you have to look, okay? I'm just gonna present a few of those here, and the opportunities exist and come every year, okay? And now exist more more and more with Brazil. This is the program that both Odair and Aline had taken advantage of. So when I first met Odair, I sent him, so we started collaborating, and I said, oh, this guy is doing some interesting things. He looks smart, and everything like that. So, and so anyway, we got a very good communication, and we work in the possibility of coming to work in my lab for an exchange for 6 months. And this program provided everything for him. And we turned out to be very good. The program offers basically everything for the students, including airfare, stipends, scholarship, and the airfare, and insurance. Yeah, you get insurance there, but somehow you have to buy some insurance here. It's kind of ridiculous. And, but anyway, and it was twice a year, the application. And I think now it's just being kept once a year. So, but every time that it comes out, I send it to colleagues here, okay? One of the conditions for this is for both undergraduate, licenciatura and posgraduação, ¿sí? Graduación y posgraduación, ¿sí? One of the conditions is that you had to be students here and go there 6 months and come back and finish here. That's the condition. You can, oh, I finished now my degree, and I'm going 6 months, and then I come back and start the maestria. No, it doesn't work that way. You have to be student enrolled in your institution to be eligible, all right? And it's a good opportunity, okay? Odair also had a very good experience because we tried it twice, and he got it twice, you know? So he got the second shot as well, and although he was on the waiting list, and we were lucky enough that he came back. So he came and graduated, okay, in 2013. The first time he came with me was in 2011, Odair? 2010, and stayed with me. Odair finished his thesis, his PhD thesis, doing this study in cactaceae, okay? So, and everything started basically with this visit that he did there, because we started working with some of the collection, plant collection that I had there in the greenhouses, and we discussed, oh, it would be interesting to look at this, to look at that, and eventually started developing the whole structure and set up a plan. So it worked out very well. We started publishing, okay, different things on aspects of reproductive biology. For instance, this is a macro and morphological first paper that we published together. And then we have the second paper on seedling morphology, something that I didn't get so much involved because it's part of his master's. So I got involved very much in the organization and the writing there. So he put me in there. And then we published this other paper that's going to be part of the talk in a little bit, that I'm going to be talking those thymomastic stamens in cacti. And this is the more recent that we have. Okay? And it's a very high impact factor journal. The three of them actually are very high impact journal that they have in working in floral reproductive biology of epiphytic cacti. Okay? Very interesting, still a whole lot of things to do. And the data, the morphological data matches very well whatever has been published in terms of the molecular phylogenies. So the clades are concordant, so which is another beauty. Aline also came in 2013, 14? 2015, and we set up a program for her, a study program to work in sugar maple. Okay, so sapling dust, she was doing, okay, what can we do with sapling dust? Because I met Aline when I came here to give a course that Odair and Sosa invited me as well. And Aline was one of the students, the postgraduate, and very, very, very bright, and always showing interest. And when this option came again, I sent it to them, and she was the one that I want to go. Okay, so we set up something that could fit within her thesis of PhD. And so she did the thesis. She just defended it in May on the Sapindaceae centro stricto in the state of Paraná. And we have this paper in review, which is about the reproductive biology of these species of maple, of acer. Okay, and fingers crossed, we'll see what happens. Okay. There is this other program, okay? Again, the government of Canada, but it's a little bit tougher. These are more difficult to get in, okay? But it's worth trying because it's also very good and it has more prestigious. Not that the others don't have prestigious, but this is for, as well, PhD students and postdoctoral fellows, okay? And sometimes there are programs for faculty as well, right? Faculty is a lot easier to obtain an exchange or a scholarship for 3-6 months, okay? So it's worth looking at this possibility. And this is from NSERC, which is the equivalent of FAPESP here, the agency, okay? And then we have this, which just definitely just came out 6 months ago, and it's a program that is more relaxed, I'm gonna put it that way, because it works between 2 faculty members. It's a dual degree, okay? It means that as long as there is a professor in this university, okay, and a professor in my university that can and want to supervise a student, okay, particularly PhD, okay, and they have agreement, okay, they can do it. So the students goes to a very easy admission path, very easy admission path. And of course, it is a committee group from the university the host university and the home university, so that the 2 of them are involved, but it's very easy to get into admission, okay? It has less restrictions in terms of languages, okay? It's another thing that is there. So it facilitates students coming with some deficiencies in English, but the condition is while they arrive, okay, they have already, they can improve and can be capable of communicate and do research and communicate in English, okay? I was gonna say, one of the conditions in this program is that the student has to have a master equivalent already, okay? If not, it has to have at least 1 year completed, let's say here, of PhD, to qualify to go there and go to TEL. Okay, that's the idea. And that's one that exists. So these are in general what we have for the university, but there are other options. And Brazil has several options as well, in which involves Canada. I didn't do a whole lot of research on that, but I'm sure you are aware that Frontiers Without Borders, I think, still works, that Brazil and has an agreement with Canada. CNPq has also some agreements to support students and fellows to visit for short periods in North American countries, including Canada. So it's, what I always think and I always say to my students is it depends on the student. It depends on the students what they want to do. Okay, if they're really motivated and they have the fire and the will to go and experience and learn, the options are there. So it's never too hard to try to get something. So is there any questions? Let me know. I can answer in Portuguese or in English or in Italiano. Or español. So my email is there in case you have a question or personal or whatever, follow up on this. And we can talk and follow up. Yes. Ajá. Solo para doctorado, sí. Solo para doctorado. Felizmente. Sí, si el estudiante no tiene maestría, es preciso que el estudiante tenga un año de doctorado aquí para segundo año comenzar con Cotutele en Canadá. Sí, cualquier institución. Sí, cualquier institución. Sí. ¿Otra pregunta? No? Okay, I'm gonna change gears and move to the next part, if you don't mind. So, you need that? So I want to talk now a little bit more of the things that I do in the lab. of the work we're doing is, as I mentioned, I mean, my life, my professional life has evolved from being a field biologist, botanist, collecting in the deserts of Mexico, went to the United States, worked in southwestern United States, all the field, all collecting plants for my master's, for my PhD, and then I started doing cytogenetics, and as a master's student, I learned taxonomy back in Mexico with Redowski, a fantastic taxonomist, and then went up to learn different things in the United States, especially cytogenetics. I was fascinated back then in the late 80s about cytogenetics, and then in the 90s with the molecular biology. So all the 90s I worked with molecular biology, but I wanted to do something more. You know, I wanted to learn about plants more and more, and why plants reproduce, and why plants have different flowers, and why plants are selfers, and what plants do not match, and why plants are hermaphrodite, or unisexual, etc., etc. So, you know, there are so many things there that I say, okay, I need to look at different ways. And the cactaceae has all that. The cactaceae has all that. So the cactaceae is a very difficult plant family, not only because it's difficult to collect. Okay, you know, when you collect mosses, you can collect 50 mosses in a little bag. When you collect cactus, you collect 1 or 2, and then you are tired and all bloody here, you know, because it's difficult to handle, okay? And that's one of my professors, the lichenology professors, told me, forget about cactus, work with lichens, you know, no, gracias. Anyway, it's just the passion that exists for everybody. Cactus always have fascinated me. It's part of my professional life. So today I'm going to be talking about 2 things, vivipary in plants, right? And the floral biology and the sensitive stamens of a prickly pear, Opuntia polyacantha, which is another plant that we have very close, basically outside my, our doors in the university, and we have in the summer the chance to work with that. So just to give you an overview of the family, for those who are in the family, you have quite a few representative of cacta members of the Cactaceae here. The family is subdivided in 4 subfamilies, okay? And the Perescioideae is the more basal ancestral, okay? Which is a leafy one, so it doesn't really look like a cactus, okay? So it's leafy. The only characteristic that it has, of course, the inferior ovary, okay, that it has, and the areoles. And then we have the other 2 subfamilies, the Maguenoideae, which is a very lonely, very few representatives. And then we have all the Opuntias, all the prickly pear, Platyopuntias and Cylindropuntians. But then this family, the Cactoideae, which basically has most of the diversity occurs in Mexico, although some of that in South America, but most of this in Mexico, includes the largest number of species, okay? So we have about 1,400 species in the family, of which perhaps 1,000 species are in this subfamily, okay? All the forms of cacti are there. And in terms of distribution, okay, as I mentioned before, that species that we're going to be talking about, the Opuntia, or the sensitive stamens, the northernmost distribution of that plant is around here. So Canada still have 4 cacti, okay? Very cold, but those cacti are very well adapted physiologically and morphologically to withstand temperatures below -20 degrees, all right? And those 2 that exist here in South America, it doesn't get as cold, but exists here in the Patagonia, okay? So it's all this. The family is American, okay? It's Americans, 100% Americana, okay? Everything that is outside the Americas has been introduced, right? It's the second, one of the 2 American families, along with the Bromeliaceae. The Bromeliaceae is the other American family, okay, unique to the Americas. These are the centers of diversity that exist in the Cactaceae, okay? And southwestern United States and central Mexico are 2 of the most important ones in North America. But much of these that exist in terms of the species are located in the South America, in Brazil, Argentina, and Peru and Bolivia. Those places are really, really, really hot spots in terms of the cactus diversity, all right? And we have, again, in terms of genetic, we have pretty much a matching situation, with the exception of the Central America and the Caribbean, in terms of some genera that do not exist in other parts of the Americas. Excuse me. So the cactuses that we have been working has several interesting features, okay? In particularly vivipary, which has been basically recorded in these places. Okay, so that's another thing that makes me switch part of my research when I first knew about vivipary in the cactaceae. I had read about it, but I had never seen it, okay? And that was in 2003. So it's vivipary and epiphytism. So I knew there was epiphytism, and we started working with that evolution of epiphytism in cactaceae because how is it possible that a family that basically lives in the deserts of Central and South America, okay, lives also in the rainforest, okay, and is epiphyte? Okay, well, those places in the rainforest are deserts, are the driest places that exist in the rainforest. So it's equivalent. So it does make sense. But the morphology of the epiphytes don't make those epiphytic cacti look like the regular siblings that exist in the mainland or on Earth, in Tierra. So this is part of the diversity that I'm talking about, that we have. So some of the Pereschia, some of the globular, columnar form. It's amazing, definitely. You are fascinated, not only with diversity in shapes, in forms, right, but also diversity of flowers. And you start thinking about who visits these flowers, okay? Why do they make these flowers? And sometimes they are all produced at the same time for such an ephemeral time, very short time for blooming, and they still manage to reproduce very well. Of course, you find very odd specimens in the field, okay? And many of these cacti that you see here, for instance, have been over-collected, okay? Because the habits are formed, some type of mutations, and they work a lot of money, right? And also because some rich people want to have these interesting plants, okay? So they pay fortunes, and they smuggle, they take. Mexico has been the victim of many eradication of plants, okay? Now things are a lot better down there. The flowers are spectacular again. So from bee pollinated, okay, bird pollinated, right, that we have a tubular corolla, and hummingbird, etc., and moth pollinated, okay? Plants can be very small from the size of the nail in your pinky, the size, like 2, 3 millimeters, 5 millimeters, is, I don't remember the name. It's here in Argentina. Losfeldia is here. It's a monogenic genus. Okay, and some others are, as I say, very, very large, up to 40 meters in height. Okay, so they are woody, basically develop a structure very sturdy, very hard. They tend to form and they be part of different plant communities, okay, like forests, dry forests, the rainforest, etc. They are basically found in every single plant community throughout the Americas. The thing is that you have to know what they are. These are the ones that tend to mix or mixed up with others that they don't look like cacti, as I'm saying. You know, they look like lianas, but it's not necessarily a liana. If you look closely to this, there are several aspects that really differentiate the presence of the areoles, particularly, because it's unique in the cactuses where the spines are. And then those spines disappear here because they don't need it in the mesic environments. They don't need the spines there, okay? In the dry areas, in the desert, they are exposed to everything, and they have to protect from the insulation, from the sun, and from predation, okay? And here, they are protected for the plants above. So they change. And these are very common here. So you are very familiar with Epiphyllophilanthus, who grows all over Maringá, okay? And it's a very nice model plant that I've been working with in my lab. And since I found it also in Brazil, that gave me a lot of material to work with. Now, just to put it in perspective, phylogenetic phylogenetically speaking, the cactus family belongs to this big group, okay, Caryophyllales. Everybody should be hopefully familiar with that. It has several characteristics, beta lines, curved embryo, etc., etc. So all this group had that. And we have a group that is called the non-core, okay, that includes very particular, very interesting features, okay. It has carnivorous plants. Carnivore is another very, very rare condition in plants, okay. Carnivore is insectivore. This is in the non-core. In the core, we have families that are terrestrial lineages from warm, tropical, and temperates, okay? And within the, oops, what did I do? And within this, we have the cactaceae, which is located here, okay? So the family, and it has, as I mentioned before, these two conditions, okay? Which are the two, basically, aspects that have drawn my directions in terms of the research. And within vivipary, we started looking also at the flower and reproductive biology. So vivipary that we have here, perhaps you had heard about vivipary. How many of you know about vivipary? ¿Conoce la viviparidad? ¿Sí? So vivipary is like us, okay? Like humans. We are viviparous because we are born alive, right? In plants, there is vivipary, okay? And I'm going to show you this video just to understand where I'm going, where I want to go, okay? There is a commercial, a little break. Okay.
A lemon shark's purpose in life isn't to terrorize tourists, it's to make more lemon sharks. And to do that, females come into the Bimini Lagoon every April. Most animals either lay eggs or give live birth. Sharks do both.
I see it.
This lemon shark has been carrying her young for about 9 months. Like baby mammals, they've been fed by a placenta, and each is joined to its mother by an umbilical cord.
The
number of babies in a litter varies between 4 and 17.
No, it's not showing it.
Delivering her babies in this shallow, safe lagoon is the closest the mother gets to actually caring for them. She doesn't give them a glance after they're born and surely wouldn't recognize them if she did. In fact, she'd probably try to eat them, although it's thought a shark giving birth is under the influence of a natural appetite suppressant, at least for a while.
The screen is split.
Okay.
So what we have to do basically. Wait. Aha, there.
Okay.
And is the one that maximizes?
I'm
not familiarized with it.
Where are you? How are you seeing that? I don't see it here. Okay.
Oh, yeah. Because
the
like
screen?
I said, yeah,
Okay.
it's
Let me
like
see.
you have two
You want to
separate
maximize? You want to make what?
Yeah.
Here. The video?
That should work. Yep. All right. Then you can.
Okay, we have to turn.
Another way you can do this is like either reproduce exact.
Carrying her young for about 9 months. Like baby mammals, they've been fed by a placenta, and each is joined to its mother by an umbilical cord. The number of babies in a litter varies between 4 and 17. Delivering her babies in this shallow, safe lagoon is the closest the mother gets to actually caring for them. She doesn't give them a glance after they're born and surely wouldn't recognize them if she did. In fact, should probably try to eat them, although it's thought a shark giving birth is under the influence of a natural appetite suppressant, at least for a while. As soon as they're born, the baby lemons need refuge and head for the shallows and the shelter provided by nearby mangrove roots.
All right, I think that's good enough.
Shark Sunday, February 10th, beginning at noon on the Discovery Channel.
Okay. Now I can go back to my PowerPoint. I don't see my arrow. I don't see my cursor. Hey. No. Oh my gosh. Okay. Nope. I don't see my icons. You see? What do we do? Nope. How do we do that? Can you come over here, please? Help. Never happened this to me. I'm sorry. And there is another video coming for you, no?
Oh, so then maybe what we can do is like reproduce the screen. Be able to do that by doing this, yes. So maybe some option.
Okay, so according to the video, so in a viviparous organism, what happened is that individuals are born alive and have an umbilical cord. Okay, and this is unique in placental animals, right? So keep that in mind: born alive and umbilical cord. In plants, okay, the first time that vivipary was mentioned was in 1966, but it's German, okay? Okay, this is germination of seeds within the fruit before the seeds are dispersed from the parent plant, okay? So at that point, there was some idea what was happening, and for instance, vivipary was described. Most of what we know of vivipary is because of the mangroves. You are in the coastline, and you have some of those mangroves, you can see that those fruits that detach, okay, eventually emerge and rupture the pericarp and start going and anchor somewhere. So mangroves are the best example for what we call true vivipara, okay, because the embryo ruptures, breaks down the ovary wall of the pericarp. There is another type that we call pseudo-vivipary that you have probably seen just presented in the margin of leaves of many, many, many plants, okay? So kalanchoe, for instance, is typical, and you can see that it's proliferating there, okay? You can see many species in the allium family, okay, that have vivipary, and that is a type of asexual reproduction that is being formed, okay? There is another type of vivipary that is even a little bit more complicated because we don't really see it. It's hidden, okay? So the embryo develops inside the fruit, right? And I'll show you a couple of examples in a little bit. So vivipary, what we know of is that it's genetic, and it's a genetic event. It's controlled by several genes that are there, okay? It's associated to recalcitrant seeds. In other words, seeds that cannot tolerate desiccant, okay, cannot desiccate. You cannot store seeds, okay, because they lost viability. And abscisic acid is basically one of the responsibles. The presence and lack of abscisic acid is one of the responsibles for the presence of vivipary. And normally, most of the cases have been associated with very stressed environments, okay? So why do I bother then to study vivipary? The vivipary has been drawn to me the attention, as I say, since I first found it and working in a collection in the Missouri Botanical Garden, for instance, and then in Montreal Botanical Garden, collecting seeds, actually, from some seeds, from plants there, okay? And it happened that I started learning that vivipary has been considered a very bad condition trait for crop cereals. And I'll show you what in a little bit, okay? So it's very rare. I'll show you why. And also, because it's rare, we know relatively very few about what's happening. And when actually vivipary happens, okay? It's unpredictable in some cases, okay? It's all about the ABA and gibberellic acid as well, some of the gibberellics. And in our case, in my case, for instance, the cases, or most of the cases that we have discovered vivipary is also present or happen to be in cacti that are in the rare list, okay? So in addition to vivipary being rare, the cacti are rare. So there are two very important factors there to why study, why combine these two data. When we look at vivipary at the commercial crops, okay, these are examples, for instance. Up above, this is wheat, trigo, this is strawberry, corn, okay? You can see very much the new seedlings emerging. This is pseudo-vivipary, that's the one in the margin of the leaves. But then in the bottom, you don't get to see those seedlings unless you cut the fruit. Only the moment you cut the fruit, you discover that those things are, and most people throw them away. No, don't. Eat them. They taste good. Anyway, bean sprouts, they taste like a bean sprouts. So this is what we call pseudo-vivipary, all right? And why it is bad? Would you guys eat some of these fruits? Perhaps not, right? It's the taste and the quality of the fruit that is being deteriorated. So in tomato, we had a tomato plant in the greenhouse, and it happened that I was there. I always go to the greenhouse almost 3 times a week. In the winter and the summer, and especially in the winter, it's very nice because it's warm in the greenhouse, and my office is very cold, right? and so we have this type of tomato, and we had 16 plants growing, and then I found this, like collecting these tomatoes and separating seeds, and opening it and say, wow. It was all like that, you know? And you can see that these seedlings were really, really, really big already, you know? And they were all very much attached, conglomerated, the radicules, but attached to the placenta of the plant, okay? So pretty much like in mammals, right? So why vivipary is happening? Sometimes that I'm thinking, and plants do that like parental care, you know, like we humans, animals, we take care of babies, right? So plants perhaps sense somehow and a response to the environment. So we have that. And this plant, I started working with that. I said, okay, what can I do, and trying to improvise. And so I started separating, documenting these seedlings, and then started growing, transplanting them, okay? And I had 2 fruits, the 2 fruits, viviparous. One fruit, I took it like this, intact, and I put it in a pot with dirt. See what happened, okay? See if the mama will take care of the babies, okay? And then the second one, I separated all those. Of course, I damaged some. I didn't recover everything, but whatever I damaged, I transplanted those individually. And those that were transplanted individually, I follow the development. So those that were planted in block like that, the whole thing, everything died. Okay? In a week, everything was gone. And of these that were there, only 3 seedlings survived to maturity, to the extent that they produce fruits again. Okay? And this is the mortality, the survival and mortality rate that we have. So keep in mind, these are viviparous progeny, all right? We're talking about viviparous progeny as opposed to orthodox progeny, all right? So of those 3 that survive, we have 3 plants. We have, again, 5, and 3 fruits that produce. And then I allow them to mature everything and open the fruits when I thought it was good to look for vivipary. I didn't find vivipary. So at least in that generation, the trait was not passed on. Maybe I needed one more. So long story short, we have pretty much that in general, there is a 1/3 of seed loss. So most of the seeds that we have in those fruits from viviparous plants are deformed, right? So what we have here is that first, you have a viviparous plant that none of those seedlings, you let them alone, are going to survive. And if you separate them and let them to grow, help them to grow, okay? Only 1/3 of that seed is going to survive. Very few of those seeds are going to survive, okay? Most of them are malformed, are deformed, are not viable, in other words, right? So this is one of the problems that we have, okay? So vivipary has been present, not only in these crops, but you happen to have vivipary in mango, in coconut, and quite a few others, in orange and chestnut, at least 20 different crops, okay? And that is a problem because the fruit is not of the good quality for commercial purposes, okay? And also you have a fruit that you had to harvest new seeds to continue growing your fruits, okay? You are losing seeds there if you have vivipary. So basically what's happening is that your fruit lowers in quality and seed set, and in general, the overall production is lower, right? In the wild, for instance, we can think about these conditions happen in the cactaceae, and we have seen it. We have done experiments with several columnar cacti back in northwestern Mexico, and we have done the same. Do not survive, okay? And the seeds that are produced from viviparous cannot overwinter, okay? Because they are not viable. Remember, they are recalcitrant, okay? They do not tolerate desiccation. They desiccate, they die. So in endangered species, this is very critical. If we don't have seeds in the wild, how do they propagate? Unless they have clonal reproduction, right? So for germoplasm issues, it's very serious. And then we are not thinking about all these issues of food security. I think it becomes more critical, okay? So how common then is vivipary? So I told you in general, gave you the example here. Let's take a look to the overall distribution. So we have, for instance, in vascular plants, okay, which we have around 300,000 species, okay? Same species, according to Elkins and Cox, okay? They have same species, further genera, and 23 to 500 families, okay? And 140 species in 80 families. So our last count, it accounts for this, that 140 species of vascular plants had been reported vivipary, which is a very low proportion, less than 1%. If you think about all the diversity that we have in plant, is almost nothing. So it's indeed a very rare. Perhaps it's more common, but perhaps we we have not studied. It's out there. It's out there. I think it's out there, but we have not looked at that. It's overlooked, okay, in many ways. In the cactaceae, as I say, we have around 1,500 species. We have around 55 species, which is a 4% percentage of the whole family. It's high, and I'll show you why I think it's high, okay, when we compare with plant families. When we see the distribution, for instance, of the families that are most of the viviparous, so as I say, we have the mangroves, okay, that are here, very common in red, and we have others in the monocots and dicot families, okay? And then pseudovivipara, prior to this, it was known in the cactaceae and this coryphantha vivipara, by the way, okay, Puntia, okay? And we have other monocots and dicot families. So, in general, vivipara occurs that we have seen in saline, dry environments. Very dry, very sparse, in flooding conditions, mareas, okay? Changing environments, and very halophilic conditions. So there are super stress. That has been correlated with changes, okay, in the production of hormonal levels, particularly abscisic acid, all right? Low levels for abscisic acid triggers vivipary, okay? And because this is what happens, so ABA controls dormancy, and it's slow to germinate. You have to have high levels of ABA in order to be, okay? And those examples that we have had basically indicate that these plants have some low levels of ABA. Some of the examples that we have are associated definitely with coastal environments, okay? But some others are not. So these are the examples that we discussed, like I mentioned about in Mexico, northwestern Mexico, all right? And we have worked also with establishment of progeny, okay? But viviparous progeny is not successful. It's like the tomato, okay? And I'll show you in a little bit. This is another species that happens to be, this is one of the species that happen to be in this region, right? And these are barrel cacti. And this is vivipary, cryptovivipary, okay? And you don't see it, again, it's like the tomato. You don't see it unless you cut that fruit, okay? And if you cut that fruit, those seedlings will never get out, okay? The pericarp is very tough. It's like a leathery pericarp, okay? And by the time the pericarp perhaps decays, it's going to be very difficult. The seedlings probably are exhausted. They exhausted the placenta, the mama, already. Those are, so the fruits are different. Not all of the fruits are like this, for instance, that have very tough pericarp. The epiphytic cacti are very particular, for instance, that have fruits that have very semi-transparent fruits that you can see through the pericarp that the fruit has some germinated seedlings there. Very, very interesting. Ripsen, this is one of the best examples. Okay, sometimes it's red, but still you can see the bulbs and the really seedlings coming out there, okay? And other species have a little bit larger, but still start emerging. So it's just a matter of looking at the fruits. Before, I didn't care too much about opening every fruit that I saw. You know, I just collected what I needed. Now I see a fruit, I had to use it. I had to check it out for that and see what happened. So we have many, many different types of fruits in which there are viviparous. This is the plant that we have been working for a while, okay? And it's distributed from Central America all the way down south to Argentina, okay? And this, when I was doing my sabbatical at USP in 2006, this plant was basically 20 meters from the entrance of the building, a botany building there with Pirani and Lucia. And then I was walking in the garden and I see this fruit hanging. And see, wow, it's interesting. So this is a plant and a fruit in which basically the first emergence of the viviparous. And they grow so much that they protruded to the pericarp, okay? And when I allowed also these fruits to develop, but the fruit never detached from the plant, okay? And also, it was the thing that eventually these seedlings started decaying, dying, okay? Because there was no nutrients. There were no nutrients. So there was nothing there to be able to do it. So again, like in the tomato, I separated and documented the morphology of those seedlings, okay? And then I transplanted in different conditions, okay? Para ver, to see this question. So they wanted to see whether ABA was involved in germination, okay? At what level was the production of ABA levels, okay? And whether those seedlings were viable or were just dead end, evolutionary dead end, okay? And how viviparous populations can maintain the cacti can maintain their populations. So these are some of the different ABA profiles that we did for the viviparous and non-viviparous material and embryonic tissue. And it shows definitely that we have low levels of ABA for that, okay? And of course, it's very difficult to catch the exact right time, okay, for what those levels are right. You discover it, and the moment things start changing, the physiology of the plant starts changing, okay? But there is some strong evidence that low levels of ABA are involved here in the ABA. So these are the experiments that set up in the Botanic Garden at USP and for the survival establishment. So we have examples that we did near the parent plant, right here. This in right there, I think that you have the plant here, and that the seedling just fell at the porophyte, and the phorophyte, at the base of the phorophyte or the host plant. And then you have here the artificial conditions, una estufa. So this is what happened. We follow first for 3 months the reproduction, okay? And you can see here the green is on the porophyte, the host plant, in other words. On the ground is the red and the control photoperiod. So it's calculated. And what we got, that basically everything was going to the same direction. Doesn't matter the treatment, everything was going to the same direction after a certain time. Another thing that we found, you see, so it's a genetic trait, okay? And water availability was perhaps one of the issues. But everything was getting the same thing, okay? Except the ones in artificial conditions. So we determined here 3 different levels. For instance, the acclimatization, the stabilization phase, okay? So if you look here, okay, this part, there is the highest mortality happen in the first month and a half. And then there is a little more of the steady, but at the end, the 3 conditions end up really dying. So is it really a good strategy to be viviparous when everything is going to die? When I first discovered vivipary in this epiphyllum philanthus, I said, wow, this is a strategy, or in cactus, in ferocactus, this is a strategy for facilitate the propagation of seedlings, very easy and faster. But now it has a serious doubt. I don't think it's very useful. I think it's a waste of energy. It's an evolutionary dead end. That's what I'm thinking, all the information. And when we put this together, this data with this tomato information, okay, which are the crops, we have a very much mirror image. So all the majority of the plants die. Any survive, okay, the seeds are not viable. So is it really good? Is it a reproductive advantage? So you tell me, okay? So I was thinking about that before. Now my ideas are in doubt. I'm confused, anyway. So can be vivipary a reproductive strategy? This is the way I used to see it, okay? So true vivipary, as we see it, okay, is putatively of sexual origin and the asexual vegetative, okay, pseudovivipary. Most of the cases that we know are in true vivipary, okay? And the difference with these two is that we have seeds that make seedlings, and eventually, if they survive, develop in adults. In asexual, those pseudoviviparous plants eventually become adults. Okay? So you have several stages going in sexual vivipary. The question here is whether these seeds are recalcitrant or not. Can they tolerate desiccation or not? And then once these plants are mature, they can either become again, pass on the character, the trait, or not, or can go in the other direction. Okay? So it's a little bit of the puzzle and see how this is working. And as I said before, in my mind, vivipary is a very rare, very interesting trait, but it still needs a lot of research. Okay? Time is the major difference that exists in the two of those in these plants. In general, as per the cactus family, I mentioned that vivipary is better known in mangroves. Okay? And within dicots, for instance, and monocots in general, the Cactaceae is the fourth plant family, okay, and the number of species in viviparous, percentage in viviparous. So it's very, very interesting, right? And then again, there are other plant families where the frequency of vivipary is very low, okay? But then again, is it really what is out there in the nature, or are there so many plant families or so many species that need to be investigated, okay? As we have published this paper, more and more reports have shown up in terms of the vivipary in several regions of the Americas as well, providing this information, which is really interesting. So anyway, where is the vivipary is happening? In the Cactaceae and many, many subfamilies. So phylogenetically speaking, okay, it's a convergent trait. It doesn't have unique features. You have an epiphytic cactus, an epiphytic, allophytic, non-allophytic, etc. So in all type of life forms. So it's very different. These are the number of species that we have. So the fruits, as I mentioned before, are so different as well into the most viviparous cactus. So what can conclude here about vivipary? One of the most important things that we can say, for instance, in terms of viviparous, and for what we know not only in cacti, but in general for angiosperms, is that viviparous occurs in basically any environment. But stress either by the lack of water, temperature, okay, and salinity are important factors, okay? The fruits are always in different characteristics, okay? And yes, we have this information about compartmentalized levels of abscisic acid, okay? And also in mangrove, a recent paper show up, okay? And then it's an expensive trait. In my mind, it's a waste of energy in general for the plant. That's what I think. The chances for survival are very low. And I'm still, so plants, I always think that plants are very smart. And I don't know why this is happening in nature. So it's weird. It's weird. So if you have any question about this part, you have to let me know. I'm getting super tired. And you have to let me know. And because the next part is with the floral morphology of the Opuntia. Any question about this? No? All right. So next time that you see a fruit, please check it out and make sure that you document vivipary in plants. You know there is more. All right. The last part is about these floral polymorphisms and Tignomasty in Opuntia. This is Denver, one of my graduate students and masters, and this is the plant that we are working with. Okay, this is Odair with Denver. That was in 2013, 2014 maybe, I don't know. Anyway, so this is the plant again, and they are collecting and doing some pollination and test. And this is a cross section just of the flower. Beautiful flowers or a prickly pear, right? The plant is very, very small, doesn't get higher than this, 20, 30 centimeters, okay, and makes the flowers. So Canada, as I mentioned, we have 4 species. This a student of mine did the distribution of the cacti in Saskatchewan. So this is where the cacti are in Saskatchewan, all the way to the south, in very dry conditions, okay, and rolling hills, as you can see here, very dark brown to black soils, okay, and in the mixed grasslands and low grasslands, so very short grasses, okay. Those are the landscapes that occur. So this is not the most distribution here in the province, and everything else is very cold, and the soil conditions change again. So limited by temperature and soil, I should say. So over the years, we have been looking at several things. Typically, flowers of cacti are hermaphrodite. That's the typical condition, and that's the typical condition also in angiosperms. Okay, hermaphroditism is the ancestral condition to be taught. All right, and we have here, for instance, not the exceptions. But as we work and started collecting and documenting samples, we started noticing different morphs that we call. Okay, so we have flowers that have all the hermaphroditic features. We have what we call transitional male, in which you have plants that have both male and female condition, but some of the tepals become staminoids, start changing to staminoids. And then we have what we call males, in which either there is no ovary or there is no stigma. Okay? So, to that condition in which the hermaphrodite condition prevails, okay, with some males present around, we call it androdioiche in plants. So this is what we have, for example, some of the floral attributes, and I'll go through that slowly. So we have here, for instance, the hermaphrodite flower, okay? In the hermaphrodite flower, for instance, we have that characteristically has the largest flowers, okay, in the larger number of ovules, okay, so, which is fine. And then in the transitional males, we have that they have larger number of stamens. They have the larger number of staminoids, of course. You can see here the serial staminoids, how a tepal start becoming an staminoid, basically forming regular, normal, up to where it becomes staminoid. And then an intermediate number of ovules it has. The males that are here have absent, so they have a style, okay? Of course, they have no, the ovary chamber might be present, okay? And some of those is totally absent, and some of those is present, but the ovules are not malformed. That's what I'm going to say. And the number of stamens is not exactly larger, but the hermaphrodite still have the larger stamen. So these are the conditions that we have in terms of the floral characters that we can measure and that we can assess. In the analysis, we had the association, most of the hermaphrodites and the transitional males that are associated by a certain number of characters, particularly with the corolla. But the males totally separate from the rest of the others, okay? And that it makes us definitely the 3 different floral morphs that we have. So it's a very interesting feature that we have not seen actually that far north in the Cactaceae, and we still are investigating what is the role of these different flowers, or is a trend towards having a change in the mating system, right? Now, the next part is about these sensitive stamens of this plant. So this plant is the same plant in which we discover that, like many other cacti in the Opuntia genus, has sensitive stamens. So here we are using a manual activation, but when insects come and visit this flower, okay, they also trigger the movement of those stamens. It's like the sensitive plant that happens, all right? And it stays, they move and concentrate in the center at the base of the style, okay? And the main stress for 5–10 minutes, and around 15 minutes, they relax again, they open up, okay? And then the insects, depending on the size of the insect, so that insect will trigger the movement. Smaller insects than that will not trigger the movement. And they can go and they take a big dip and shower and pollen, etc., and then go, okay? So we have several videos, and here we document the insects come and take off and touch the stigma lobes. So we assume that this is one of the conditions in which, one of the strategies in which plants attract pollinators. So the response of plant parts to a stimulus is not uncommon, okay? So you know some of the examples typical in the leguminose, okay, the mimosa, okay? But existing fungi, algae, bryophyte, ferns, germplasms, and basically the whole plant kingdom. So, but it's fungi not plant kingdom, okay? Carnivorous plants are the most interesting that exist in terms of triggering and having these particular electrical signals, okay? So we had the ionea, okay, and we had the sundew that we have basically changes in hormones that are triggering the secreting glands of the sundew, for instance. And in here, the electrical charge of the ionea basically trigger one of the fastest traps that exist, okay? Here, in the mimosa that we discussed or that I mentioned before, there is a change in turgor pressure in the cells, and that's what causes the extensor to close and move the leaves to the closure. So there are many mechanisms that exist in angiosperms, in this case for the response. In the cactaceae, we know that exists in this family, subfamily, in this genus, Brasiliopuntia, Cylindropuntia, and Opuntia. Only on those three that we know of, okay? And in the Portulacaceae and Portulaca, okay? And then we have these other recent reports that exist in the Lamiales. So they are distributed throughout the lineages. So we started looking at these stamens, okay, and try to find out what made this movement, what caused this movement, okay? So we knew that there was a turgor mechanism, changes in cell pressure, et cetera, cell walls, action potential in the cell membranes, okay? And we started looking at the different portions of the stamen, okay? And we brought to the lab. And we look at the microscope, okay? There was Choi, the postdoc from Korea, and he looked at the upper part, and we started discovering different types of papilla, okay, protuberances that exist there. In the tip, there were small papilla with foldings. In the middle, there were epidermal folding, but the number of papilla increased in number and size, okay? And here at the base, they were even bigger, okay, and in size as well. So there was something happening. We look at other species, and we find pretty much the same. So these are some of the same group of the Opuntia that we're discussing, and we look other groups outside of the Opuntia in which the stigma, or the filament is smooth, as opposed to have this papilla that in those species that have the sensitivity, right? So I say, well, it has to be that. We continue looking, and then we have in confocal microscope. We look to see what was the structure of those cells, okay, to look from inside. This is not the best, but it's another slide. So at the tip, the cells are long and narrow and folding, so enhancing the surface area, okay? In the middle, they're longer and narrow that I have as well, okay? They have the foldings, but the number of papilla increases in number on size. So most of the movement has here. And at the base, they are longer, the cells are longer and wider, okay, with increased number of papilla. Why? Because the moment that this filament is moving inwards, okay, you need flexibility here. You need longer cells and less wider cells, so you can move inwards in that way. I'll show you in a little bit. This is what we have. The intermediate size that we have here, the base, have the wider and longer cells, and intermediate in the middle. So you need to move the base, okay, in the other part and reach the tip. This is the same study of the confocal microscope, and you can see here the characteristics of those cells, okay? So which provides some of the elasticity that is making that, or facilitating the movement of that filament. Odair did the characterization of the anther, as well the filament, I'm sorry, and he found also that the cell walls are very thin. Okay, it's another characteristic. And that's a facilitator of moving inwards. And the papillae are unicellular, okay, and where layer covered external perineal wall of pericrament. So these are basically the thin single cell walls are the sensitive areas that occur in these species. These are the sensitive, these are the non-sensitives with thinner walls here. And these are the papillae that they have, the unicellular cells. So the filament in general has several layers that discover, and we still have to see what are the components of the calcium oxalate. There are very much some of the components in cellular movement. So just to make this story shorter, this is what we have. It's basically the same movement or the same mechanism that trigger sensitivity in other plants, okay? The tactile protuberans have thin epidermal walls, okay? And that facilitates the movement, triggers the movement, the sensitivity of this species. So in this part, we're still working. Denver is still developing part of the job. So my conclusions are inconclusions because I don't know still what's happening. We're still looking at that, and we want to look at another type of microscopy, and hopefully we can do more of the analysis in terms of the composition, the chemical characterization that exists there. So with this, I just probably want to finish. A lot of people have worked with me, and these Brazilians here, Adeusa and Auri, were very important in developing the hormone information in USP. Marcia, as I mentioned before, other graduate students from the past that were here, and then, of course, all the new ones that are in the past one as well, and others coming that are here. So a lot of people, it's not, my job is a teamwork, and sometimes we are unhappy, but sometimes we're very happy, and most of the time we're happy. And one of the most beautiful things is that the relation continues, some of the people. So after this, I can take any question if you have any question. Thank you. I'm sorry for making it so long. Yeah, it was long. Very. Any question, anybody? Yes. Okay.
É fascinante pensar nessas coisas, né? Obrigado pela apresentação. Existe alguma relação nessas opuntia entre a movimentação dos stamens e o fato de ter flores masculinas ou hermafrodita?
No lo sé exactamente porque no. We have not done, sorry, I can't think in Portuguese. We have not done the correlation in that particular point. You know, we're looking now and trying to characterize the filament, and the movement. So these 3 species that we have investigated in the Opuntia, they do share that anatomical characteristic, but we cannot look at the correlation of floral morphs with the sensitive response. But it's a good point. Maybe those other 2 species that we look at, nothing has been reported in terms of different floral morphs. All of the flowers are hermaphrodite in those 2 species that I mentioned. And the other thing is that, remember, Opuntia has a very long distributional range. So in my view, I think that the population up in Saskatchewan might be different from a population that might be happening in northern Mexico, in terms of the flower morphology, the floral morphs, Maybe selection is acting more strongly in the northern and with the climate in Saskatchewan that is in northern Mexico that is not so cold, for instance, and they don't have the pressure for pollinators or anything like that, and the plant can propagate much easier. So, but yeah, it's a very good point.
Bom, mas de qualquer forma, a movimentação dos estamens deve ter, eu imagino que vocês imaginam que tem a ver com polinização.
Sim, sim, absolutamente, sim, absolutamente.
E qual seria as ideias sobre qual a vantagem disso?
See, the basic idea is that they help in pollen transfer, okay? The other idea is, so while protecting the pollen, okay, or touch, coming to the center facilitates the transfer of pollen to the stigma because it's closer, okay? The other thing that we have noticed recently is that the pollen is bimodal. There is a very tiny pollen that seems not to be fertile, and the good pollen seems to be in the middle, in the center. So if that's the case, then the good pollen is protected by the longer anthers and offering cheap pollen to the pollinators and saving the good pollen in the center. So the pollinators that crawl under there, they do collect the good pollen and then again and take the touchdown from the stigma or land in the stigma in that regard. Yeah. Sí. Tem varias ideas.
O sucesso reprodutivo dessas espécies, ele não está muito bem estudado, porque como rapidamente essas plantas são cobertas pelo gelo, então os frutos e as sementes não têm tempo de ter sucesso de germinação e formar novos indivíduos. Então essa reprodução sexual dessa comunidade, então ela ainda não está muito bem descrita e a gente não entende muito bem como que está funcionando, né? Ela está se propagando lá vegetativamente. Então seria interessante ter uma comparação com populações de áreas mais perenes, né, como o México, onde a mudança da sazonalidade seja mais constante, para poder ter um comparativo e ver o real papel dessa interação entre os animais, né, e também ver esses morfos florais, principalmente em relação ao movimento do estigma, né? Porque, de fato, uma planta que está ficando masculina, esse movimento de estigma, ele teria, como o Hugo disse, proteger um pólen mais fértil e deixar à mostra o pólen barato para aquela abelha que vai coletar o pólen e ela sair ela se alimentar do pólen barato e levar o pólen, de fato, viável e uma outra planta. Então, eu acredito que deva ter taxas de autopolinização, porque quando o comportamento do polinizador, ele passa por todo o androceu e quando ele deixa a flor, ele passa pelo gineceu. Então, um pouco de autopolinização acontece. Então, tem um percentual aí de autopolinização que eu acredito que aconteça nessas plantas. Isso se evita, exato. Mas, Hugo, você tem alguma informação da biologia reprodutiva dessa espécie em outras regiões, como no México?
Não. Temos dados, por exemplo, de Denver ha fatto la polinización cruzada manual con bolsa. La mejor es la polinización cruzada. la más efectiva. Y cuando no es selfer, não funciona. Não funciona, tem que ser polinização cruzada.
Então, ela tem um mecanismo de autocontrole, né, para evitar isso. Mas, de qualquer forma, eu acredito que ainda haja uma taxa de autopolinização ali que ocorra nessas espécies. Mas, no final das contas, ainda, esse set fruit, né, a frutificação que tem lá, o sucesso, ele ainda é questionável, porque as plantas jovens não têm tempo de recrutar, né, o recrutamento lá não funciona. Então, ali é um bom exemplo da dinâmica da natureza, né? Ela está no limite da sua distribuição e está ali, né, tendo resposta ao ambiente de uma maneira que a gente ainda não tem uma visão muito boa.
Alguma outra questão? Não?
Ok.
Iva?
Sim.
Mas você tem mais braço maior.
Se você puder responder, porque eu não vou saber.
I can listen.
A viviparidade, eu achei interessante o termo viviparidade em planta, mas não seria apenas uma quebra de dormência precoce por causa da queda do nível de ácido abscísico, de ABA? Low levels of ABA can be the seeds grow up.
Low levels of... I'm sorry, I didn't understand the question. Can you repeat?
Em espanhol.
Espanhol? Trata espanhol?
Sim.
Não será que... Repete para mim.
A viviparidade.
La viviparidad.
Não seria só uma germinação precoce da semente?
¿No sería una germinación precoz de la
otro
semilla?
Es otro nombre que se le da a la viviparidad, germinación precoce. Se le conoce exactamente lo mismo. Sí.
Los bajos niveles de ABA.
Exactamente.
Por los bajos niveles de ABA.
Es exactamente lo mismo. Sí. Se usa como término alternado. Viviparidad de germinación precoce. Sí. Sí.
Obrigado.
Sí.
¿Y hay alguna similaridad con el término para la pseudo-viviparidad con el brotamiento de las hojas en Kalanchoe, por ejemplo? ¿Hay una diferencia entre que yo diga brotamiento o pseudo-viviparidad?
Es porque es asexual, o sea, no está envolviendo nada de las células reproductivas. Sí. Entonces es todo sexual. Es una separación como lemna, prácticamente.
¿Y ya se ha visto algo de eso en el cactus acá en el Mandacaru?
No.
Cereus, me parece.
Cereus
ilmana.
Sí, no sé.
Muchas gracias.
No, de nada.
Alguma outra questão? Pergunta?
Quase animais, né?
São
quase animais.
Sim, são quase animais, sim.
É uma similaridade, porque a nossa viviparidade também é uma germinação precoce do ovo.
Sim. É interessante porque tem as mesmas características. É cordão umbilical que está na placenta da planta mãe del óvulo, da placenta da mãe no fruto. Sim. O funículo é cordão umbilical. Sim.
Algo mais? Não? Então...
Obrigado. Se algum de vocês tem uma pergunta ou está interessado em conhecer ainda mais da universidade, mande um e-mail. Está a pergunta para a maestra Lídia ou o maestro Luiz.
O professor ele vai ministrar uma disciplina de biologia reprodutiva, né, Souza? Junto com o professor Odair, o Souza, o professor Hugo, né, que vai ministrar a maior parte, na próxima semana, segunda, terça e quarta, no período da tarde. A Estela ela enviou para as pós, né, comunicando quem quisesse se matricular também, tá? Então ele vai estar até quarta-feira, né, professor?
Sim.
Até aqui. Se tiverem alguma dúvida, quiserem entrar em contato com ele, né, pode nos procurar lá no PGB e aí a gente entra em contato, vê um horário, né, para conversar também, né, posteriormente com mais calma, de repente.
E lembra que eu não sou o único professor da universidade, no meu departamento é grande, tem tanta diversidade de especialidade. Allora.
Correto? Então, vamos agradecer ao professor
Grazie.
pela belíssima palestra.
Grazie.
Obrigada.